Hello guys, welcome to my YouTube channel Let's Learn Pharmacy. Yes, so today in this session of our lecture 3 we will discuss about the excretion, and excretion and pharmacodynamic properties. And after completing the pharmacodynamic we will end this general pharmacology. After general pharmacology we will go through the MCQs of the general pharmacology, previous year MCQs and some new more MCQs which may help for your examinations. Now first, what do you mean by excretion? So you know that major route of excretion is the kidney. Major route, the drug what get excreted, no, that drug is excreted majorly in kidney. Glomerular filtration does not depend on the lipid solubility. You have to understand this. You know that in the excretion majorly there are three processes. One is glomerular filtration, tubular secretion, tubular reabsorption, and the third one is a tubular secretion. Now here glomerular filtration, it doesn't depend on the lipid solubility. Means it own depends on lipid soluble, whether the drug is lipid soluble or water soluble, it doesn't depend. Why? Means the glomerular filtration is there, no, it allows or it filters both lipid and water soluble. Glomerular filtration, it depends on ppb and renal blood flow. ppb means it depends on the plasma protein binding, whether the drug is bonded with plasma protein or not. Plasma protein and renal blood flow. For that reason our blood is in colorless, sorry, not blood, our urine is in colorless or some or yellowish color or pale yellowish color. Why? Because these glomerular filtration removes the blood components from the urine. Yes. Now next, tubular reabsorption depends on the lipid solubility. This reabsorption, tubular reabsorption means that the tubes which are present, no, in the nephron tubes, what you called Henle's loop, descending, ascending, collecting tubule, these all are the tubular reabsorption which it depends on the lipid solubility. That is, if a drug is lipid soluble, more of it will be reabsorbed and less will be excreted. If the drug is a lipid soluble, then the excretion of that drug is very less and absorption is more. Then if the drug is water soluble, means the excretion is more excretion and the absorption is less. Then in acidic drug poisoning, this is very important, asked many times in your examination. The acidic drug poisoning, that is a salicylate, barbiturates, chlorpromide, methotrexate, these all are the acidic drugs. Urine should be alkalized with sodium bicarbonate. If these acidic drugs causes poison, means then what we have to give? We have to give the alkaline drug like sodium bicarbonate. Then basic drug poisoning means some basic drugs they cause poison like morphine or amphetamine. Then urine should be acidified with the ammonium chloride. We have to give ammonium chloride through IV route to maintain this level, to bring down the basic drug poisoning. Next, tubular secretion doesn't depend on lipid solubility or ppb. This won't depend on the both. Clear? Then half-life. You know that what is this half-life? Half-life, simply understand, it is the time required to reduce the plasma concentration to the 50% of the original drug. How much time taken to reduce the 50% drug? 50% drug, that is half-life. If metabolism is more, half-life is less. Simple. Why? Means metabolism, it will break down the drug, more breakdown of the drug, so half-life will be less, very less half-life period. More metabolism, why it occurs more by metabolism? Because of plasma, because of the not bonding of the drug with the plasma protein. If the drug binded with the plasma protein, then there is a metabolism is very less, so half-life is also more. If a drug follows a first order kinetic, its half-life is constant. If the drug, if it follows first order kinetic, means then the half-life is, that is a constant half-life. Next, pharmacodynamic properties. Till now we discussed about the pharmacokinetic property, that is action of the drug, action of the action of the body on towards the drug, what our body show reaction to that drug. Okay, now here what drug will show reaction to the body for pharmacodynamic. This is the study dealing with the effect of the drugs. What is the effect of drug on the body, like mechanism of action, okay, mechanism of action, side effects. These are all of the pharmacodynamics, that is the drug, effect of the drug. What's the therapeutic effect of that drug on the particular human. Then see the receptors, these are the binding sites of the drug. Receptors are the binding site of the drug with functional correlate. Receptors play a vital role here. These are the binding site for the drugs. Receptors help to play a vital role in binding. They will collect the drug, they will bind with the drug so that to get activated and show some therapeutic effect. Next, some of the effectors are there here. The first one is a G protein coupled receptors, that is a GPCR. It is also called a metabotropic receptors. These are heptahelical receptors, means these receptors contain 7 receptors in this, 7 transduction membranes. Okay, these are heptahelical receptors. That is the G protein coupled receptors has 3 subunits. How many units? 3 subunits, that is alpha, beta and gamma. When all 3 joined, G protein is inactive. If alpha, beta and gamma they are present all together, then this G protein it is in inactive form. Clear? Inactive form. If this gamma if this alpha gets separated from beta and gamma, then your G protein coupled receptor is in active form. Then ionotropic receptors, it is the second type of the receptors. The drug binds directly to the receptor, okay, located on an ion channel without mediation by G proteins. Means here drug directly goes and bound with the receptors, and these are fastest acting receptors. One of the fastest acting, like example GABA, NMDA, 5-HT3 receptors. These are some examples for the ionotropic receptors. Then enzymatic receptors, you know that enzymatic receptors also very important. Insulin, growth hormone, prolactin, cytokines, they act via enzymatic receptors on the basis of enzyme. Next, our topic is a dose response curve, that is a DRC. What is this dose response curve? It is the graph between the dose of a drug. It is the graph. We have to plot a graph, no? That is the graph between the dose of a drug administered and effect produced by the drug. When we administered the drug, okay, that and effect, what amount of effect is produced, means how much concentration is there that drug, that and also the dose of drug administration. These two difference we are studying. Difference, that is a dose response curve. Then therapeutic index helps to measure the safety of the drug. Therapeutic index, you know that therapeutic index, it is very important to know the safety. We have to carry this therapeutic index. Lethal dose counting, very important one. Next one is of our new drug development or novel drug development. See, before developing of the drug, means what we are using in our market, no, many drugs are available in the market. How they have arrived in the market, how they get approved, okay, those drugs which are distributed in the market. Now for an example, you take Dolo 650 or that Vic Section 500 or Saridon, etc. These all drugs, before releasing to market, they should under drug discovery phase, preclinical studies and clinical trials. Without these 3, they are not introduced in the market. No, first they have gone this drug discovery, then some of the preclinical trials, then clinical trials. After that clinical trials, we are introducing such drugs into the market. Then drug discovery phase. See, most new drugs are discovered through random screening, compound oriented, target organization, target oriented or rational drug designing. Many drugs discovered, most of them are randomized, means they are discovered by means of screening only, and some by compound oriented. Compound oriented means when 2 or more molecules when we are adding, no, they will show some of the therapeutic effect, that one type of the drug get introduced. Simple. Like this way the drugs are invented. So now preclinical studies. This is the first preclinical studies before leaving to the market. This is very important one, preclinical studies. Here the lead compounds, means here drugs are tested on animals to know the whole pharmacological profile. In preclinical studies we are not using the human volunteers, we are using here animal. Okay, we test are first preferred on small animals like mice, rat, guinea pig, etc. Guinea pig, etc. First we will prefer always small animals. After, if these small animals holds good, then we go for large animals, like you take dog, then some of the other animals, deer, etc. Yeah. Next, clinical trials. In this clinical trials, you know that more 4 major phases are there: phase 1, phase 2, phase 3 and phase 4. These 4 phases helps to study the pharmacological efficacy, then side effects, some of the toxic effects, okay, which these 4 clinical trials. Thus, in the phase 1, the drug is tested in normal human volunteers except children. Here first what we have to do means we have to test the drug in the normal human. We cannot determine efficacy in this phase, very important one. Here in this phase efficacy is not determined, only we are knowing the toxicity of the drug or the pharmacokinetic property of the drug. This is to understand the toxicity and pharmacokinetic studies, where in clinical trials. Here we are not discussing about the efficacy, we are not discussing about the therapeutic use. No, just only we are discussing here toxic, how toxic is the drug and what is the pharmacokinetic study. If we given this drug to the normal healthy human volunteer, means if he holds this drug for how long, that good healthy human volunteers can holds good, that is very important here. Okay, very important. Here patient is not treated. No. For in first phase we are not giving for the patient, we are giving for good, means he is healthy human volunteer. This point remember you. Then some drugs like anti-cancer drugs can produce unacceptable toxicity, and we cannot expose healthy humans to such a toxicity. Means some of the anti-cancer drugs are there, they will produce horrible effects. So to prevent that horrible effects, only in except in anti-cancer drug, we are selecting patients in clinical trial, that is phase 1. But except this anti-cancer drug, remaining all drugs we are studying on the healthy human volunteers. Phase 1 trials for anti-cancer drugs are done in the patients, yes, then conducted on 20 to 100 volunteers. In phase 1 trials always we are referring small members, 20 to 100 members. Then phase 2, the drug in this phase is tested in small number of, that is 20 to 200 patients. Homogeneous population, means same genus. We can determine both efficacy and safety in this phase. In this phase 2 we can determine the efficacy and the safety of that drug, and this is the first inpatient study. First inpatient study means we are studying this drug in the patient in the phase 2. Purpose, what is the purpose of this phase 2? To establish therapeutic efficacy, dose ranging and ceiling effect. Then in phase 3, here the drug is tested in large number of patients at several centers to include patients with different genetic makeup. Here we are studying the patients by giving the drug with a huge amount, like 5,000 population, different genetics. We are using different genetic makeup. If a drug is approved, then drug is marketed. If here in this phase 3, if that drug is approved by the FDA, Food and Development Association is there, no? If it approved means then we are going for post marketing, or that is phase 4. Here therapeutic conformity is done, where in the phase 3 up to 5,000 patients from several centers, that is from several heterogeneous population, we are treating under this phase 3 trial. That is, next is phase 4. It is the post marketing surveillance of a drug to know the rare adverse effects or those occurring with prolonged use of the drug. Some of the anti-diabetic agents or anti-psychiatric agents or anti-hypoglycemic or some of the anti-hypertensive drugs. So these are used for the long time. Now by giving these drugs, is there any kind of a toxic effect? What are the rare toxicological effects on the human? Okay, on this phase 4 we are studying. Large number of patients here being treated where in the phase 4. Yes. Now one more point, recently phase 0 trial has been also added, and it is known as the micro dosing studies. This is added to expedite the marketing of the drug. Phase 0 trial, recently it was discovered. Why? The main intention of phase 0 trial is to accelerate the marketing of the drug. To speed up the marketing of the drug, we are using this phase 0 trial. Then schedule of the drugs, some of the schedules are there. Okay, that is framed under the Cosmetic Drug and Cosmetic Act and Drug and Cosmetic Rule 1946. C and C1, that those are biological and special products. F1 that is bacterial and vaccines. G, caution, it is dangerous to take this preparation except under medical supervision. You go for the market, you take any cough syrup, there will be scheduled G, scheduled H. What are these? These are the precautionary measures, or these are the quotations given. Auxiliary label, what we are telling. No? H, drugs that must be sold by retail, that is schedule H, sold by retail only when a prescription by RMP, that is a registered medical practitioner, is produced. X is, these are the psychotropic drops requiring special license for manufacture and the sale. Yes, here we completed the first unit, that is general pharmacology. Okay, I want to complete this whole unit. Why? Means because this play a vital role, that is the general pharmacology, vital role in your exam also and for your knowledge purpose also. Basic thing you have to understand in this general pharmacology. Next we will go our important session that ask many times in GPAT examination, that is cardiac diseases drugs that are given to treat the heart problems. Clear? So thank you.